First detection of biofluorescence in a deep-sea anglerfish. Biofluorescence has been observed in a variety of fishes, but is rare in deep-sea environments where light from the surface cannot reach. Here we document biofluorescence in an oceanic anglerfish, the Pacific football fish. Green biofluorescence was observed in small spots on the distal surface of the esca. While the wavelength of bioluminescent light is unknown for this species, it is possible that light produced by this species also results in biofluorescent emission that may create a more complex lure for attracting prey or mates. Animals display an extraordinary variety of colors and behaviors that have evolved for a multitude of purposes, including crypsis, warning coloration, mate attraction, and thermoregulation, among others. Additionally, organisms across the tree of life have evolved the remarkable ability to bioluminesce, or create their own light. While colors, behaviors, and bioluminescence are components of optical animal displays that are easily documented, perhaps one area of biological display that is understudied, in terms of breadth and scope, involves patterns or colors that can be difficult to see with the naked human eye. Biofluorescence is the ability of an organism to absorb visible wavelengths of light through the use of proteins, organic pigments, metabolites, or minerals, and re-emit that energy at longer wavelengths. While biofluorescence may be difficult to see in the full light spectrum, it is particularly prevalent in the marine realm, where longer wavelengths of light are quickly absorbed in the water column as light penetrates deeper, resulting in a uniform blue light spectrum, 470 to 490 nm. The prevalence of blue wavelengths of light at greater depths excites fluorescent proteins and organic pigments in organisms, while reflective colors are subdued due to the decreased amount of ambient longer wavelengths at these depths. Biofluorescence in marine fishes is taxonomically widespread and is found in bony fishes and sharks. Additionally, it is observed across various habitats, occurring in shallow tropical coral reefs, species that inhabit sandy or rubble habitats, and Arctic fishes. Green is the most common fluorescent emission in fishes, although yellow, orange, and red also occur, and some species will fluoresce multiple colors. Biofluorescence is also found in species that occur at greater depths, where the blue light spectrum may be limited, precluding ambient fluorescence from occurring. Deep-sea fish fluorescence may be due to shared ancestral characters with shallow water relatives, or it may be paired with another source of blue light, bioluminescence. Bioluminescence is the production of light by an organism, generally involving luciferin and the catalyzing enzyme luciferase. In fishes, it is thought to have evolved at least 27 times independently, Davis et al. 2016. One diverse and fascinating group of bioluminescent fishes is the deep-sea anglerfishes of the ceratioidae. Of the 177 recognized ceratioid species, most mature females possess a bioluminescent organ, the esca, that is at the distal end of a modified dorsal fin, spine, the elysium, Peach 2009. While there has been considerable work since the 1870s describing the bioluminescence of ceratoid anglerfishes, reviewed by Peach 2009, the occurrence of biofluorescence in these fishes has not been explored. On 7 May 2021, a single female Pacific football fish, Himantiophilus sagmius, Lophiformes himantiophilidae, washed ashore at Crystal Cove State Park in California, USA, Figure 1A. The specimen was collected by park staff, frozen on site in a -20-degree freezer, and later transferred to the Department of Ethology at the Natural History Museum of Los Angeles County. Prior to preservation, photographs were taken using a Canon EOS 80D DSLR camera with a Canon EFS 18 135mm lens and a Canon EF 100mm macro lens. Fluorescent images were taken using a DFP1 dual fluorescent protein excitation flashlight, Night Sea, that emits both royal blue, 440 to 460 nanometers and green 510 to 540 nanometers light. An appropriate corresponding long-pass filter for each excitation color was placed in front of the camera lens for all fluorescent photography. Faint green fluorescent emissions were detected in a spotted pattern on the distal surface of the ESCA when using the royal blue fluorescent excitation light, figure 1b. The ESCA also emitted green light. However, this may be due to autofluorescence of bioluminescent compounds remaining in the ESCA and not actual fluorescence. No other fluorescent emissions were detected on any other part of the body or in any other excitation wavelengths. Figure 1, a unique anglerfish, a female Himantopholis segmius prior to preservation, A. The scale bar represents 50 millimeters. The ESCA under royal blue excitation light, B. The scale bar represents 15 millimeters, and the interior of the ESCA is to the right. Both images are of the right side of the fish and have been reflected. With the exception of the Colophrenae and Nectaridae, female ceratoid anglerfishes possess a bioluminescent ESCA, Peach 2009. There is significant structural and morphological variation in the ESCA among ceratoid families, and even between closely related species. This is apparent among the 22 recognized species in the Himantophilae, which have been described as having the most variable ESCA in terms of size and shape among all ceratioid anglerfishes. Metamorphosed females of H. sagamius display an intricate bioluminescent system with a large main ESCA, distal escal appendages, and bright silvery tips on all other escal and elysial appendages. Haneda, 1968, made the only observations of a live specimen for this species that washed ashore and was kept alive in an aquarium for 8 days. In addition to noting that the species can expel a large plume of bioluminescent material when agitated, he observed that all three features of the bioluminescent system, ESCA, distal escal packages, and distal ends of other appendages, produce light, but noted that the distal tips of the escal and elysial appendages only luminesced when agitated. In addition to the ESCA, a number of secondary structures have been observed or postulated to produce bioluminescence in ceratioid anglerfishes. These structures include the hyoid barbel of Linophrine species, which does not use bioluminescent bacteria to produce light, the caruncles of the Ceratidae, the tip of the second dorsal fin spine in Cryptopteras, Monk and Herring 1996, and teeth of various species, Beebe 1926, albeit the latter two have never been confirmed to luminesce. Interestingly, Beebe and Crane 1947 also reported numerous secondary sources of bioluminescence in their description of H. azurelians, including the bases of the bucklers, patches of skin on the dorsal and ventral portions of the caudal peduncle, and directly anterior to the anal fin, on the rays of the caudal and anal fins, and on rough areas of the chin, snout, and elysium. Secondary luminescence has been corroborated in Himantopholis by observations of glowing skin on the dorsal portion of the caudal peduncle, and in rays of the dorsal and caudal fin in H. albinaires 1961. However, luminescent patches of skin or fin rays were never noted by Haneda 1968 during his observations of H. sagamius. For fluorescence to occur in deep-sea environments where surface light is limited or completely absent, a secondary source of light needs to be present. While bioluminescence and biofluorescence are widespread, the combination of the two in deep-sea environments is rare. The most notable fish example involves stoplight loose-jaw dragonfishes, Melicostia species, which produce blue bioluminescence in a post-orbital photophore that excites red fluorescent emissions from an accessory orbital photophore directly under their eyes. This red emitted light is not visible to potential prey and is thought to aid in predation. In cnidarians, the combination of biofluorescence and bioluminescence has also been observed in a species of fish-eating deep-sea siphonophore, a renna species, which has tentacles that terminate in a red bioluminescent bulb that is surrounded by fluorescent tissue that emits red to yellow wavelengths when excited, and it is also present in crystal jellyfish, where blue bioluminescence excites a green fluorescent protein, producing green fluorescence. Bioluminescence in Hemantophilis may provide enough light for fluorescence to occur if that light is of the correct wavelengths to excite fluorescent regions. Unfortunately, the wavelength of light produced through bioluminescence in Hemantophilis is unknown. Haneda, 1968, did not state the color of light that he saw in the only live observation of H. sagamius. Beebe and Crane, 1947, observed a brilliant turquoise blue and also flashing yellow light from H. azurlicans, and Christopher Collini observed a white light from a specimen of H. albinaris. These observations reveal inconsistent luminescent colors for species of Hemantophilis. However, bioluminescence is predominantly constrained to blue-green wavelengths. If luminescence in H. sagamius is blue, then it is possible it could excite the regions of the distal surface of the esca that emitted fluorescence in the specimen examined here. Fluorescence in the deep sea could function as a more complex and enticing lure in ceratioid anglerfishes, while bioluminescence among ceratioid anglerfishes is primarily thought to aid in attracting prey. It also could be important for mate recognition. Males of Hemantophilis are not parasitic, and a complex luminescent and fluorescent display from females could aid in attracting males. Deep-sea fluorescence may only be relevant to those species that have the ability to see fluorescent emissions. However, invertebrates, such as mantis shrimp, and a variety of fishes have been shown to be able to view these wavelengths. It is also possible that escofluorescence is a shared trait among lophiform fishes, as a fluorescent esca has also been observed in Antinaria striatus, Shaw, 1794, a shallow water frogfish, DeBrouwer and Hobbs, 2016, Peach and Arnold, 2020. However, we feel this is unlikely as the frogfish emitted a red fluorescence, unlike the green fluorescence observed here, likely involving non-homologous fluorescent proteins or pigments, suggesting an independent origin for fluorescence. To our knowledge, this is the first observation of biofluorescence in a bioluminescent ceratioid oceanic anglerfish, and we hope that future observations will reveal fascinating, complex systems of biofluorescence and other deep-sea bioluminescent species.